Did you know that there's a peptide that your body makes that's encoded in DNA that has nothing to do with your dad? You know, that guy that you haven't seen since he left to get cigarettes when you were 5? And as logic would hold, if you didn't get it from your father, that can only mean... That's right, my lab rat legion. At long last, we finally have a peptide that lets me combine my two loves of early 2000s hip-hop references and modern biohacking. Oh, and it might just crank up your basal metabolism up to Super Saiyan 4. So, buckle up. Today, we're talking MOTS-c. All right, Ronick, let's do this. Now, MOTS-c's full name is actually Mitochondrial Open Reading Frame of the 12S rRNA type C, but that name is almost as thick as your mom, so we're just gonna stick with MOTS-c. But the molecule itself is genuinely one of the most strange and interesting compounds in therapeutic biology. It's 16 amino acids long and made by your mitochondria, which are these little cell-like structures, aka organelles, that live inside your actual cells and have both their own genome and an evolutionary backstory as a free-living bacterium that got swallowed by another cell 2 billion years ago. More on that later. But when you exercise hard enough, these aforementioned mitochondria fire off this unique chemical messenger that waltzes into your cell nuclei and tells their DNA to hurry up and get to work. So of course the internet found out about it, and now people are running 12-week cycles that they learned about on TikTok. And on July 23rd, 2026, it's going in front of an FDA advisory committee that may or may not give it a path back into legal compounding pharmacies. So today, we're gonna do what we always do. We're gonna figure out what MOTS-c is, what it does, what the evidence says, and then what people are actually doing with it. What's up, guys? I'm Dr. Alex, board-certified urologist, fellowship-trained men's health specialist. And on this channel, we take whatever the wellness and biohacking communities are obsessing over and run it through the filter of actual published medical literature while making jokes that only your inner 7th grader can appreciate. But first, smash the like button, subscribe for the rest of our upcoming peptide deep dive lineup, and drop a comment down below. Have you actually run a MOTS-c cycle? Are you considering it? Or did you just show up because mitochondrial peptide sounds vaguely like a DBZ episode plot point? All three reasons are perfectly valid, and remember, we read every comment. I mean, I do. Ronick would, but I'm pretty sure he's cheating on me editing a video for somebody else right now. Let's go. First off, what is MOTS-c? As we mentioned earlier, MOTS-c is a 16 amino acid peptide that your body makes naturally, and one of the things that make it particularly unique is where it comes from. Most peptides like BPC-157, TB500, growth hormone releasing hormone, and basically all the ones we've covered on this channel so far are coded in your cell's nuclear DNA. That's half from mom, half from dad. Basic 7th grade science textbook stuff, minus that cool looking S we somehow all managed to learn how to draw before YouTube was a thing. Believe it or not, MOTS-c is not coded there. MOTS-c is coded in your mitochondrial DNA, which is a completely separate genome that lives inside the mitochondria themselves and which you inherited entirely from your mother. Yeah, believe it or not, mitochondria are inherited solely through maternal genetics. Now, MOTS-c was discovered in 2015 by Dr. Chang-In Lee and Dr. Lee Pinchas-Cohen at USC and was first published in the journal Cell Metabolism. The PMID is 257384459 if you want to read about it. And Ronick will throw up the citation on the screen for the handful of you that still remember how to do that. And what they did was scan the mitochondrial genome for short, open reading frames. These were chunks of DNA that could code for peptides that nobody else had noticed, and tucked inside the 12S ribosomal RNA gene in a region everyone assumed was purely structural, they found a sequence that coded for this little 16 amino acid peptide. They called it MOTS-c, started injecting it into mice, and well, weird things began to happen. First off, fat mice on high-fat diets stopped getting fat, and insulin-resistant mice got insulin-sensitive, and old mice started behaving metabolically like young mice. It was a heck of a debut, and it single-handedly kicked off the entire field of mitochondrial-derived peptides, or MDPs. MOTS-c was the first one shown to circulate in your bloodstream and act on tissues far away from where it was made. A hormone produced not from information stored in your cell's nucleus, but by this separate weird organelle. And speaking of which, we need to talk about these mitochondria things a little bit more. Quick disclaimer, if you actually talk to girls in high school and therefore don't find ribosomal genetics thrilling, you can go ahead and skip to the time code Ronick is definitely going to remember to put somewhere down here. Now, for everyone still with me, let's kick off this biochemical bus ride.
Seatbelts, everyone!
Mitochondria are the organelles inside your cells where you make ATP, which is the molecular currency your body uses for literally everything. You have 100 to a couple thousand of them per cell depending on the tissue. Heart muscle, packed. Liver, packed. Skeletal muscle, standing room only. But here's where things get weird. Mitochondria actually have their own DNA, their own separate circular DNA that looks shockingly similar to bacterial plasmid DNA. They have their own bacterial-looking ribosomes. They reproduce by binary fission. And the leading theory is something called endosymbiotic theory, which was that mitochondria were originally free-living bacteria. The theory goes like this. Scientists' best guess is around 2 billion years ago, two bacterium got into an argument, and then one bacterium resolved said disagreement by swallowing the other one. It failed to digest it, eventually somehow worked what must have been a terrible stomachache, and instead, the two of them figured out how to get along. They then eventually developed into what we now know as the eukaryotic cell, which is what every plant, animal, and fungus on Earth is built of today. So mitochondria are basically a Gotenks level energy merger that just kept going. Now, the second weird thing about mitochondria is how you inherit them. Nuclear DNA is pretty simple and intuitive. You get half from mom and half from dad. Mitochondrial DNA, on the other hand, come entirely from your mom. Every mitochondrion in every cell inside your body was originally inside your mother's egg. The few sperm mitochondria that snuck in during fertilization were actually tagged for destruction upon entry, which means that you and your siblings share identical mitochondrial DNA, which is the same as your mom's going back hundreds of thousands of years. It's so stable that geneticists can map every human alive today back to a common female ancestor in East Africa about 150,000 years ago. A mitochondrial Eve, if you will. Or Lilith, if you happen to be a vampire. Shout out to anyone who played OG Vampire the Masquerade back when gateway PCs were still a thing. Anyways, mitochondrial DNA codes for 37 genes in total, all related to energy production machinery. And for 50 years, we thought that's all it did. But the discovery of MOTS-c blew that entire framework up. It turns out that the mitochondrial genome also codes for signaling peptides that travel outside of the mitochondria and sometimes outside of the cell entirely. It can then go to other parts of the body and tell it how to behave. So mitochondria aren't just power plants. They can actually spam the email inbox of every other cell in your body. And that's what makes MOTS-c a mitochondrial peptide, not just because it acts on your mitochondria, but because it's made by your mitochondria before eventually being released into circulation. Now, moving on to mechanism, MOTS-c basically does two main things. First, MOTS-c acts as a retrograde signaling molecule. That means that under stress, like exercise, fasting, or hypoxia, MOTS-c is able to leave the mitochondria, pop into the main cell nucleus, and then it can start telling your nuclear DNA which genes to turn on via transcription factor known as NRF2. In particular, it amplifies genes that control antioxidant defense, fat burning, and mitochondrial biogenesis. Now, mitochondrial biogenesis is just a fancy way of saying make more mitochondria. But what's happening is that a feedback signal from your power plants is now able to tell Central Command, hey dude, we need reinforcements down here. Build more of us, and while you're at it, burn fat, not sugar. This is genuinely new biology that is barely a decade old. 20 years ago, back when I was in college as a biochem major, we didn't think that mitochondria could talk back. The second thing MOTS-c does is that it activates AMPK. AMPK is like the master switch for cellular energy. When AMPK turns on, your cells burn glucose, oxidize fat, and shut down expensive things like fat storage, and a lot of things can activate it. Metformin activates AMPK, berberine activates AMPK, and hard exercise activates AMPK. MOTS-c activates AMPK by inhibiting an enzyme in the folate cycle, which causes a metabolite called AICAR or ICAR to build up, which then kicks AMPK on. MOTS-c is basically using your folate cycle as a pull cord to start the AMPK engine. There's also a tissue-specific kinase interaction with CK2 that flips it on in muscle to prevent atrophy, yet flips it off in fat to drive fat burning. We have the same molecule with opposite effects in different tissues. It's the molecular equivalent of Jekyll and Hyde, but one that works to burn fat and preserve muscle. I cannot overstate how fascinating this is. We're talking about a unique strategy for lean tissue preservation independent of the androgen axis. And the net result is this. MOTS-c essentially acts as an exercise mimetic. It produces in cells the same molecular signals you get from doing actual cardio, which of course is the exact hook that got biohackers and myself interested. Anything to avoid cardio, huh, Ronick?
Cardio is the devil.
And if we want to talk results, at least in animals, the data is genuinely fascinating. Mice on a high-fat diet treated with MOTS-c almost completely prevented diet-induced obesity. They had the same food intake and displayed no appetite suppression. They just raised their basal metabolic rate, burning more energy, and then converting more white fat into the metabolically active brown type that exists to generate heat. In type 2 diabetes models, MOTS-c restored insulin sensitivity and protected pancreatic beta cells. In bone loss models, it prevented osteoporosis. And going back to its antioxidant effects, in a traumatic brain injury model, it crossed the blood-brain barrier and reduced neuroinflammation. MOTS-c single-handedly improved outcomes in obesity, diabetes, cardiomyopathy, osteoporosis, and brain injury. In mice. Always in mice. Now, MOTS-c does have strong correlational data in humans. Your endogenous MOTS-c levels drop with age, and they drop even further with metabolic disease. And there's even a genetic variant called K14Q, mostly in Northeast Asian populations, where MOTS-c is functionally broken. And men with that variant have a significantly higher risk of both sarcopenia and type 2 diabetes as they age, unless they exercise a lot, at which point the metabolic disadvantage disappears. And it's one of the cleaner pieces of evidence that anyone has ever produced for the idea that exercise is medicine built right into this molecule. So, animal data, incredible. Human correlational data is strong. Human interventional data, however, it's a little thin. Now, there has been one real human trial. A company called Cobar developed a stabilized MOTS-c analog called CB4211 and ran a phase 1 trial in 23 obese patients with non-alcoholic fatty liver disease. And the drug worked. Sort of. It dropped the liver enzymes ALT and AST significantly versus placebo. But the liver fat measurement, the actual endpoint everyone cared about, came in essentially identical between the drug and placebo. The trial also got hammered by persistent injection site bumps. Cobar tried to reformulate it, but then it ran out of money and consequently dissolved in 2023. And you guessed it, the CB4211 program died with it. And as a result, there has never been a successful phase 2 or phase 3 trial of MOTS-c or any analog. The pharmaceutical pipeline is essentially a graveyard with one sad solitary tombstone in it. Which leaves us, as always, in the gap. We have promising preclinical data, a strong human correlational connection, and a broken pharmaceutical development. We're once again building a house on a foundation of rat studies and hope, and complete disregard for our own personal safety, but, we'll get there. So, what do we do about this? Well, obviously, we turn ourselves into the lab rats and start shooting it up in our kitchens. Obviously. Which leads me to this big disclaimer. What I'm about to describe is what the biohacking community is doing. This is not my recommendation. I'm not telling you to inject anything. I'm a board certified urologist and I've grown quite attached to my medical license. I'm just reporting on what's happening because pretending that it isn't happening really doesn't make anybody safer. It just makes them less informed, which is what this channel is trying to fix. That said, here's what people are doing. The most recent circulating protocols for MOTS-c is somewhere between 5 and 15 milligrams injected subcutaneously 3 times a week for 8 to 12 weeks, followed by 4 to 8 weeks off. The logic goes like this. MOTS-c has a short plasma half-life of 1 to 2 hours, but its downstream effects on gene expression last way longer. Pulse and rest dosing lets the signaling cascade play out without constantly hammering the system. It's kind of like how growth hormone works, but on an even longer time scale. Now, I have seen more aggressive protocols where people are running 1 to 3 milligrams daily, but biohacker forums are full of variations. And most people are actually combining this with SS31, which is another peptide that we're going to cover soon that's actually designed to help with mitochondrial repair. But not now, because Ronick has already told me that this script is way too long. Common reported benefits include improved cardiovascular endurance, faster recovery, and a mild fat loss without the normal muscle loss that you'd expect from a deficit. And common side effects include the same injection site bumps that Cobar had, heart palpitations, insomnia if dosed late, paradoxical fatigue in the first week, and hypoglycemic-like symptoms in patients who are already fasting or low carb. We haven't seen anything acutely dangerous in published case reports, but no long-term human safety data exists. 0. We don't know what daily MOTS-c does to a human over a year. We've just never done the study. But I'm sure someone watching this video has. Now again, this is just what people are doing, not what I'm telling you to do. I'm just a glorified penis mechanic with published expertise in PEDs and biohacking, and I am probably not your doctor. Although I did just recently learn that a lot more of my patients watch my videos than I thought. Hi guys. Most of you are late in your labs. Now, if you're considering taking this, please talk to a real physician who can monitor you. And if you're sourcing from a website with more discount codes than clinical references, please factor that into your risk assessment. So where does this stand legally? Well, it's the same place as the rest of the peptide universe right now, an media-hyped dumpster fire. You can watch our recent peptide update video if you want the whole regulatory breakdown. But for the sake of this video, just know that the FDA's Pharmacy Compounding Advisory Committee, the PCAC, is reevaluating MOTS-c for compounding eligibility at their hearing on July 23rd and 24th, 2026 at FDA White Oak. MOTS-c is on day one alongside BPC 157, KPV, and TB 500. The committee is reviewing MOTS-c specifically for two indications, obesity and osteoporosis. They'll hear testimony, review the data, and then vote on a recommendation. The FDA isn't bound by the vote, but it seems to follow it most of the time. In practice, even with a maximally favorable July 2026 outcome, you're looking at least several months before licensed compounding pharmacies can legally produce MOTS-c for patients. Notice and comment rulemaking takes at least 60 to 90 days. So, as I said before, July is a milestone, not the finish line. One more thing worth mentioning. WADA, aka the World Anti-Doping Agency, added MOTS-c to its 2026 prohibited list. Section S4.4, banned at all times in elite sport. So if you're a competitive athlete subject to drug testing, MOTS-c is not a gray zone substance. It is completely banned. And if you're like the rest of us and not competing in elite sport, a WADA ban usually means something's working. And you know who else isn't getting drug tested? Any athlete competing in the enhanced games, which, believe it or not, your boy is going to be attending live. Ronick, I am so stoked to see Mitch Hooper and Thor go head to head on deadlift. I wonder if they're going to let me touch the bar afterwards. Now, for my honest take, MOTS-c is one of the most genuinely exciting molecules in metabolic research right now. The biology is wild. We're talking about a peptide made by an organelle, encoded in a separate genome inherited entirely from your mother, that's able to walk into different cells' nuclei and tell your DNA to act like you exercise more than you do. Guys, this isn't just biohacker hype. This is textbook rewriting biology. The preclinical data is broad, deep, and reproducible. The mechanism is elegant, and the correlational human data is consistent. But so far, the only human interventional data is one failed clinical trial. Not because MOTS-c didn't work, it just didn't happen to work the way the trial was hoping it would. And we still don't have any randomized studies showing that injecting MOTS-c into healthy adults safely improves any clinical outcome over a meaningful time frame. And we have no safety data past 28 days, which means we don't have any dose response curves, and it leaves weird theories like, could it react with antibodies that then react with your endogenous mitochondrial signaling, just totally out in the ether. And like I've said before, promise and proof still live in different neighborhoods, but if anecdotal reports are to be believed, this is probably one of the most exciting peptides on the modern biohacking horizon. So if this video helped you out, smash the like button, subscribe for the rest of the peptide deep dive series coming up, and check out our videos on BPC 157, KPV, TB 500, and GHKCU, the regulatory update, and the GLP biologic addendum. We've done a lot of videos lately. Drop a comment and tell me whether you're running MOTS-c, considering it, or just showed up to learn what a mitochondria is. I read every one. To the three of my urology buddies who actually watch these videos, see you in DC at AUA later this week. Or last week. I have no idea how far behind Ronick is in his editing. I'm Dr. Alex, and as always, stay curious, stay skeptical, and proceed accordingly.